the bacteria leaves a signal, a chemical signal that the neutrophil chases, but at the same time, it faces all these conflicting signals that come out from the other cells that it finds in the environment. So if the world is also complex for the, and has many different and conflicting signals also for the single cells, that if we follow this logic, even the cell needs to have some form of a memory or being able to predict or anticipate what are the events that are going to come up in the environment and be able to respond accordingly. But we have two different levels of organization, where on the one hand, the mouse uses a neuronal network, which is can do highly sophisticated computations. But on the other hand, the cell only has a network of genes and proteins. So how can both of these networks be able to achieve this seemingly similar type of computations? So if we look in neuroscience, how we try to explain this behavior is that we have a very complex external world, then the neuronal network makes a form of internal model. So it extracts important principles or statistics from the environment, and it uses this internal model to be able to causally guide the behavior. But if we look in classical cell biology, how we think about the cell, well, we think that cells are very simple machines that only associate a stimulus with a response. And in my opinion, this has come about because in cell biology, the experiments are usually done in very simple environments. So you stimulate the cell with a constant signal. So the cell only sees one bit of information. The signal is either there or not there. And if my environment is constant, then I don't require any memory or any learning to be able to live in this environment. Also, if you take the other side, if your environment is completely random, again, memory and learning is not going to do any good because there is nothing to extract from the environment. So only if my environment has some forms of regularity in terms of the signal, then also the biochemical network, in this case, the network of proteins that processes this information from the environment, requires some form of memory, signal integration, or learning in order to be able to deal with the world in which it lives. So the question that we are asking in my group is, on the one hand, how complex are cellular computations? So what is everything in terms of computations that in particular protein interaction networks can do? And if we are able to extract some form of dynamical principles for these computations, we would like to see on a theoretical basis whether these rules of computations also scale up and they also be applied on the level of neuronal networks, and is there any similarity between these biological computations that happen on different scales of organization in living systems. So from theory, the generic framework of how we see computations, as we've heard several times yesterday and also today, is this framework of attractor-based computing. So we have any network, it can be neuronal or biochemical. In mathematics or in theory, we usually understand how the dynamics of this network or each of these nodes changes over time. And we have many theories, so we can calculate the overall, what are the possible solutions of this network. So as Ulrike also showed yesterday, we can have these stable fixed points that are separated by saddles or unstable fixed points. You can have more complex, like oscillations or chaotic solutions and so on. In general, how we think about the computation is that the system it goes over time and it's attracted to one of the stable solutions. So what matters are only these asymptotic or long-term stable solutions in the network. Hirsch and Barth nicely formulate this for neuronal networks, but the same is how we think about biochemical networks in single cells, is that as the system evolves over time, when there is signal from the environment, the system switches from one to the other stable state. And this is what we know or we term as a computation of the network. And then each of these states is associated with particular behavior of the system. So we wanted to understand whether this framework of computation actually also applies to biochemical network with a very simple question. And the question was, how do single cells sense signals when these signals change over time? So The cells do this by these transmembrane receptor proteins, where the chemical signals from the environment bind, the proteins get activated, and the whole biochemical networks gets activated. And this determines then the response properties of the network. So we worked in particular, we chose one of these receptors. The epidermal growth factor network is a typical proto-oncogene, and a few years ago, we identified the minimal, what are the most important components in these protein interaction networks that regulate the dynamics. Because we have the detailed biochemical knowledge of this network, we can also understand its dynamical properties. So this network, maybe you can already see, it has a double negative feedback and autocatalytic activation. So from theory, this is classical, can produce bistability. In this system, this depends on the EGFR concentration or this receptor concentration on the membrane. So if you have, for certain concentration of the receptor on the membrane, the system can be either in a basal EGFR phosphorylation state or in a high protein activity state. So both solutions will be stable. If we think into this landscape, it will be a landscape with two wells. One well is the basal activity, the other well is the high activity state. So what would it mean if the system will be organized or if cells have EGFR concentration that correspond to this bistable organization? Then it's very simple to predict with the simulation that if you give it even just a single pulse of a growth factor, the system will switch from the basal to the high activity state. You remove the pulse, the system stays in this state because it's a stable attractor. So in a way, the system has a form of a permanent memory, which means that if you add other signals in the system, the cell is not going to be able to respond because it's already in the active state. So it doesn't care anymore if there are other signals that come in the environment. So we decided to test this also experimentally. So the experiments that we do is that we place single cells, and we work with epithelial cells in general. We put them in microfluidic devices where we can do these complex signals with any different frequency from the outside. And then we have fluorescently tagged our proteins in the single cell so we can record both their localization, but also their activity. So we did perturbation experiments. As I said, as we know the network topology, if you overexpress the receptor or you knock down one of the other components, we ensure that the system is exactly organized in this bistable regime. So when we stimulated the cells with 5-minute chemical signals every 30 minutes, what we see is very similar to what we saw in the simulation. The receptor activity increases with the first pulse. So you switch from the basal to an active state, and then the system stays in an active state, irrespective of how many signals the system sees. Which means that if my world is dynamic and I have a long-term memory, which comes from stable attractor, that the system is fixed into a stable attractor, then the cell is not going to be able to integrate signals, and it's not going to be able to respond to changes in the environment. So it has the robustness property, but it does not have the property to be able to flexible and respond to other signals. And this is actually the case for the oncogenic situation. So in the most of the cases, EGFR mutations are typical for breast cancer. And in the most typical mutation is that the receptor is overexpressed. So the system is exactly poised either in this bistable regime or on the other side in the high activity regime, which means that the receptor is continuously active, it's continuously signals, and cell continuously proliferate. So this is not the general, or it's not optimal way how cell would function and be able to respond to signals, chemical signals that change in the environment. But this very simple experiments also showed that there are limitations of our thinking of that the system with signals from the environment just introduces switching between attractors, because we see that this cannot explain how cells process signals when these signals change over time. So very similar to what Ian just showed you, we propose from the model that what would be optimal for the cell is actually not to sit in this bistable regime, but to sit exactly at this critical transition. In this case, it's a different type of a dynamical bifurcation. It's a saddle node bifurcation. But if the system sits close to this transition, so if it has EGFR concentration that correspond to this regime, this would correspond to ability of the cell, or in this case, the receptor, to activate. When you remove the signal, the system will stay transiently active. So it will have a memory, but it will be form of a transient or a temporal memory. And then the receptor activity will reset, which would allow the cell then to activate with every new signal. So basically, the cell has both the property of a stability, because you maintain your active state for a transient period of time, but you also reset. So you're again responsive to new signals that change in the environment. So when we measured the activity of the protein in single cells, this is just an example from a single cell. When you give a growth factor pulse, the protein activates. When you remove the pulse, protein stays active for a transient period of time, and then it resets. And this memory is actually on the order of 40 to 50 minutes on average. So if you think about the underlying phosphorylation, dephosphorylation process, this is sub-minute. And this memory is on an order of hour or longer, which means that the system gains an additional long time scale in which it can keep the information that it has seen the signal before. And the duration of this memory does not depend whether you stimulate 5 minutes or you stimulate for 1 hour. The duration of the memory is an intrinsic property of the system. So we have shown that receptor network, so I show you the example here for the EGFR, but we have shown it that this is a typical property for the whole family of receptor tyrosine kinases, not just for this molecule. They seem to have a signature of critical organization. So they sit very close to this dynamical transition. And because of this, they have this emergent property of having a transient memory. And this transient memory comes with an additional add-on to the system that it gains this slow timescale. So basically now you have two different timescales that the system has, and it can use potentially for computations. And this is only because the system is poised such that it sits close to this dynamical bifurcation. So then the question was, of course, why cells would need a memory. So there are many cases in our bodies or in any system where cells actually have to migrate over very large distances in a complex environment, so among many cells. So this is an example from development, from germ layer formation in zebrafish. And if you have seen the video or you watch the video carefully, you see that the cell migrates in this direction. Potentially, it will get confused, so it will go downwards, but eventually it resets and it continues on its path upwards. So if I put this into a cartoon, this means that I am a cell and I need to move in a long distance. And this is the case, for example, during development, but the same problem arises also during wound healing. So you have to move along a very crowded tissue. You are guided only by local signals or chemicals that the cells are secreting. These signals will be noisy, they will disrupt it, they appear and disappear. So the typical migration pattern of cells is that they migrate in a random walk migration pattern. When there is a signal coming from one side, they will polarize and move in the direction of that signal. So if I move in a directed manner, only when I see the signal or when I sense the signal, and I start in a random walk immediately when the signal is disrupted, then cell will never be able to achieve this long range migration. However, if the cell has a memory, okay, I moved in this direction, my signal disappeared for a while, but I will continue moving in the same direction, then cells can actually bridge these periods when the signals are disrupted. But also this memory, very similar to the questions I was just asking, if another signal appears while I'm in the memory regime, then the cell should be able to sense this, and it should be able to redirect its direction of migration. So again, it is this property of balancing between two opposed features. You have to have robust migration to be able to bridge this disruptive and noisy periods, but you also need to remain flexible to be able to adapt to new changes in the environment. So we put the same cells now in microfluidic devices where we generate gradients from the chemicals from the outside. We make these gradients dynamic, and we wash them out after a while. So the gradient comes from the left. The cell will polarize. It will start moving in the direction of the gradient. When we remove the gradient, the cell continues moving in the direction of the gradient, and again, for a range of 40 to 50 minutes on average. And when it loses this memory, it will start to move into a random walk migration. So cells need this memory in order to be able to continue migrating in the direction of a previously seen signal. However, this memory would be very important if you move in a complex environment. So if you have a signal that comes from the left and it's disrupted, if the cell again starts moving immediately into a random walk migration when the signal is disrupted, then it's not going to be able to do one long range migration. So we did experiment where we put the signal from the left, we disrupt it, but when still the system is within this memory range, we reestablish the signal from the same side. Then we disrupt it again, and while the system is still in the memory side, we switch the signal from the opposite side. So we wanted to see whether the cell is actually able to integrate all this information. So it's indeed what the cells do. They start moving to the left. We disrupt the signal. They have a memory, so they continue moving in the same direction. We disrupt the signal again, and we switch it to the opposite side. And even when the cell is in the memory range, it's able to sense actually that, oh, now I don't have a signal on this side, but I have now a signal on the other side. So the cells repolarize and start chasing and the signal on the opposite side. When we switch off the third signal, the cells still have a memory of that signal. And when they lose that memory, then they start a random walk migration. And this is a form of a working memory that is very comparable to the working memory that you have in a nervous system, because you store information over time, but you remain responsive to other signals. So we argued here that cells also use a form of a molecular memory to be able to navigate when the signals in the environment continuously change. And this is important exactly to be able to achieve this balance between robust migration when your environment is changing and noisy, but also be able to flexibly adapt exactly to these changes. And we thought, okay, this is if cells have a basic form of memory, can one actually also look for properties of learning on the level of single cells? So how to think about learning in biochemical networks, because they're not equivalent to neuronal networks. We don't have the synaptic changes and plasticity in the connectivity. All our connections are well defined, but this is also not a new problem. There are experiments, and this is one of the, I think maybe even the first experiment. It was done by American physiologist Jennings in 1907. He took a single cell organism, which is called a stentor, which is this shape like ciliate. And he showed that if you stimulate the ciliate with beads, the ciliate experiences or displays a sequential pattern of well-defined behavior. So if you stimulate it with beads, first it tries to bend away. If the beads still keep coming, then the second step, it will reverse its cilia to try to kind of spit out the particles. If this doesn't help, it will contract, and eventually it will detach. So it goes to this really defined sequence of behaviors, which shows that they can modulate their responses depending on a prior experience. In addition to this, he has also shown and that center can exhibit a very basic form of non-associative learning, which is habituation. So under different conditions, when you stimulate the ciliate with pulses, in this case, again, it was just temporal addition of these beats. What the system does, if the beats are not harmful to the system, they're just annoying, it will, the center will respond with a decline in its response. So at some point it decides, oh, well, it's annoying, but doesn't harm me. So it stops contracting, actually. And this experiment has been very recently recapitulated in the group of Jeremy Gunavardena in Harvard. And this has also sparked a series of theoretical work. Okay, do we have a biochemical or minimal biochemical network motifs that actually can exhibit these basic forms of learning, such as habituation? But in this class of very basic forms of learning, there is almost kind of the opposite, and that is sensitization. And this is the case that upon a repetitive presentation of a stimulus, the system actually shows an increase in the response properties. So this is the example, like if you rub your hands, they will start getting warm, and you'll start get a pain, but after a while it goes away. And this is because you're repeating this stimulation, the nervous system gets activated, so you get this increase in response, you get the pain, it lasts for a while, but then you lose it. However, even in as much as we tried to look, we didn't find that there is a mechanistic model that explains how sensitization happens, even for a neuronal network. And sensitization is very interesting because it has been also very often being discussed also from an evolutionary perspective, that it is a mechanism that supports survival. So if you have a signal that repeats into the environment, potentially you think, oh, this is probably harmful, so you increase your responsiveness in order to be able to properly survive in this environment. So we thought, okay, we take our single cell network, and let's see, or epithelial cells that we work with, and let's see whether they have these basic forms of non-associative learning. And I'll show you the example of sensitization, but we have also looked at habituation. So as I mentioned, sensitization has this particular feature that when you repeat a stimuli, you see a response increment, but this is not everything. Also, if you withheld the signal for a while and you give it again, then the system shows the same amplitude of response. But if you withheld even for a longer time and you present the stimulus, the system has reset it. So it's a transient process, and you have to go over the same sequence of behavior to relearn the behavior. So if this would be a neuronal system, we would think, okay, I have some statistics of my signal in the environment. The system needs to form an internal model to extract the information about the environment and generate this complex behavior. But in this case, we only work with single cell. So our hypothesis is that the internal representation for a single cell comes through the transient dynamic or these dynamical states in the biochemical network, and the cell uses these dynamical states to be able to extract causal influence, and in this case of morphological behavior. So the most simplest behavior that we could think is changing in the shape of the cell. So we do the experiments where we stimulate the cells, again, in microfluidic devices with the growth factor pulses with some duration. We record the protein activities in single cells to see how the biochemical network encodes this. And then we also quantify the shape changes with respect to these responses. So this is the examples of the cell. When you see, I hope you can see, when you see some green flashes, this is actually when we deliver these pulses, and you see that there is this continuous change in morphology. If we quantify that change in morphology, so we do 1 minute pulses with 3 minutes interpulse interval, what we see when we look at the change in the perimeter of the cell shape, we see that we have this increase in the response, or this amplitude of the morphological change, and they respond in a form of an oscillatory pattern. When we deliver it with the last pulse, we see that the cell maintains this type of strong change in morphology for a transient period of time, and that it resets. So you have these basic forms that correspond to the non-associative learning of a response increment, a form of a temporal memory, and the system resets. And if we look at the protein dynamics, in this case, we measure the activity of the receptor at the same time, we see the similar features also in the internal representation of the cell. So you have this climbing activity in terms of the activation of the receptor. You see this transient activity, which is maintained, and then the system resets. And this is not everything. We also have seen that the system actually has even more complex features, such as frequency sensitivity, meaning that more frequent stimulation results in a more rapid and more pronounced response increment, which is also, or well, slightly just in the opposite way, is one of these hallmarks of habituation that have been defined, but actually no one even defined response properties for sensitization. So this is one of the properties of sensitization that we were able to derive from single cells. Even though this is a very simple example, I think it demonstrates that even biochemical networks in single cells can have very basic forms, in this case of non-associative learning. So to us, being primarily coming from theory, it was important, of course, to understand, okay, what are the features in these simple biochemical networks that actually lead to the memory and learning. So I mentioned this memory comes because the network is organized close to this dynamical organization, dynamical bifurcation. And we know from dynamical systems that if the system sits close to a saddle node bifurcation, what happens when you give the signal? So you're actually changing this landscape. This landscape is not fixed. So when you start here, the system has only one stable steady state, so one well. When you give an input, this landscape changes. So the system goes to the high activity state. When you remove the signal, you change back the landscape to the one well of the basal activity state. But because you sit close to this dynamical transition, you have an area in this landscape where you come down from three sides and you go away only from one side. And in this landscape, the surface is slightly tilted. And because it's almost flat, you're going to go slowly on this surface. And this is what in dynamical systems is known as a ghost set. And it's kind of a ghost. You had an active state. This state is not stable anymore, but the system keeps kind of a ghost of this active state. So you start moving down this shallow slope, and this slows down. And this is why the system gets this memory, and it gets it on a timescale which is order of magnitude longer than the timescale of the underlying processes, and then the system will fall down. Basically, it will reset. So in this system, it was, of course, easy to identify because we worked with a minimal network motif. We knew or measured also experimentally all of the parameters, so we can model it in details. But how would you identify it if you have a large scale network? So we have recently done this kind of generalization of these ghost sets. So basically, for any network, you can define a form of a Lyapunov function, which you can, in this case, we used kinetic energy of the network that you can minimize. And if the kinetic energy is 0, this is going to give you your fixed points. But if the kinetic energy is close to 0, it's going to give you areas in this landscape which are relatively flat. So it's going to be able to identify this range of this ghost attracting sets. And because the kinetic energy is close to 0, the system is still, you can linearize it, so you can locally calculate the eigenvalues of the system. And if you look at the maximal eigenvalue, this maximal eigenvalue has a gradient from negative to positive, which is what directs the flow. So if you have the trajectory of the system will directly go or pass through these ghost sets. So what these ghost sets help is, despite that the system is away from any steady state, they're enabling to direct the flow in the system and how the system will develop over time. So since this is not formally a stable state, of course, the question is, well, is this stable with respect to noise? Because what we observe in cells is, of course, that biochemical networks are inherently noisy. So would we still be able to observe this quasi-stable dynamics even if we have noise in the system? So what we did in the simulation is we increased noise intensity and we estimated the trapping time of the system in these ghost sets. So for minimal models, you can do this even analytically, but what we saw is that this trapping time, or how long you spend in this ghost set, so how long is the memory, is actually very stable up to very high noise intensity. And from the experiments, both from the EGFR and we also measure other proteins in the network, we were able to estimate the noise. And this is, of course, does not only represent, it's a mixture of the technical and the biological noise, but we are between 10 to the minus third and 10 to the minus second in the experiments. And this is probably also why we were able actually to observe this memory in the single cell measurements. But the most important message is that networks that sit close to these dynamical transitions, they also they could work with such ghost sets that enable the emergence of these long transients, and these long transients are robust to noise. And this is one important characteristic, that if you would like to do any sensible computations, of course, the dynamical objects with whom you do the computations, they need to be robust to noise. So how does the learning happen? Well, again, it is a mixture of the system sitting close to this saddle node bifurcation. As I mentioned, this gives the system a slow time scale, but also very important to remember the fact that when we have dynamic signals, what happens is that this landscape continuously reshapes. So when the system is organized at the bifurcation, it has only a single fixed point, and that is the fixed point here. But what happens when we give the stimuli in the simulation, you see that this number, so all the squares are the different fixed points in the system which appear and disappear. And what you get is this kind of a funnel of slow points. So the system starts moving along with the signal because it wants to reach the high activity steady state, but you remove the signal, so you reshape. The trajectory starts going or tries to go back to the fixed point, but it moves on this set of slow points. And because it's slow, when the other signal comes, it does not reach the fixed point. When the other signal comes, it is pushed forward. So actually it can generate this climbing activity. So this set of slow points, it's despite that giving the memory to the system, it also enables to have this climbing activity upon repetition of the signals. So this organization close to a dynamical bifurcation is important for cells because it enables them to sense time-varying signals because they get the form of a dynamic memory. But also this slow dynamics is important for the system to generate a climbing activity and thereby have this basic form of sensitization. And very important to mention here is that during this whole process of the integration of this signal, the system actually maintained away from a steady state. So the computation is formally done with the transient of the system. So we have argued, or we would like to argue, that we also need to change the mathematical framework, how we study networks that are stimulated with time-varying signals, so that we need to switch from non-autonomous system description, where we only define the properties of the system through the network topology and number of steady states the system has to a non-autonomous system description where the environment or the external signals are part of the system. Because as I showed you, when our signals change over time, the number and the position of steady states can change. So they formally don't even represent anymore the solutions of the system, but you can show that there is a family of solution operators where the solution of the systems are now the trajectories. So formally, the trajectories of the dynamical system will be the solutions and how we need to describe the response of dynamical systems when they are subjected to time-varying signals. So considering that we have found these ghost sets, or that cells use these ghost sets for having a memory and also basic forms of learning, we went on to more formally define these ghost sets. The definition is very similar to what Ulrike has shown you before, that we have a ghost attracting set if it's a close bounded set that does not contain any semi-trajectories in forward in time. You can define a basin of attraction of this ghost attracting set where the trajectories enter, they spend some time, and then they potentially leave. But if you define it like this formally, then it's very easy to see that you can have more complex structures of these ghost attracting sets in phase space. So basically, you can align them such that the system goes on one of these ghost sets, stays for a while, then falls on and goes to the next ghost attracting set. So basically, you can generate these channels of ghost sets, or you can loop the trajectory back and you can generate a cycle of these ghost sets. So what would be the dynamics, for example, of one such a ghost cycle? So we did very simple geometric models in this case, but you see exactly the same. So the trajectory of the system stays on one of the ghosts and very rapidly transitions to the second one. It will stay for a transient period of time, and then it will transition to the next one and so on. So you get a very reliable sequential switching dynamics, which is also maintained even when you have increased noise. Yes.
So this is it's similar to like a hop, like hopping between attractors to generate like a massive stable behavior.
The properties will not be equivalent because here, how long you stay in one of the attractor is a property of the ghost set. In that case, if you're just with the noise, you cannot control. Yes, so this is, let's say, I would like to argue that for computation, this is reliable in contrast to if you only rely on the noise of the system.
Is that noise level or noise properties, right? Because there could be some property of the noise. Right?
No, right. So we have only done this, or also how robust the system is. It's just an additive and Gaussian white noise, nothing else.
Just to note that might be
interesting. Yes. So this is just exactly what I just said. The system stays on the one ghost and hops to the next one, that hops to the next one. So you can go on in a cycle forever because they are arranged in such a way that they will always switch between the different ghost attracting sets. You can quantify this. So for example, the period that you're going to get on this ghost cycle is very reliable, up to very high noise intensity. The trapping in the ghost vicinity is also very reliable, which comes from the property of a single ghost set. And very importantly, for this sequential switching dynamics, and we heard some talks yesterday as well, is that you maintain your behavior stable for a transient period of time, but then you have a very rapid switching to the next. And we see again that this difference between long trapping and very rapid switching is also maintained and is not disrupted with the noise. And to us, this resembles very much this sequential switching that you see in neuronal ensembles. This is just one example. That there has been a range of experimental papers. This is an example of encoding of different tastes in the rat gustatory cortex, where they measured the neuronal activity. And then you see that for each of the different tastes, what you see is a sequential switching activity pattern. So when you repeat the same stimulus, what happens is only that you can you can have a slight variation when the system goes to the next sequence, but the sequence and the order of the sequence remains the same. And for the different days, the encoding is by different sequences. So this suggests that a neuronal information encoding, especially for such sequential switching activity patterns, could potentially happen through Gauss scaffolds that are generated in the neuronal network. So we wanted to test this a bit more. So we wanted to test what are the computational capabilities of these Gauss scaffolds. And what I forgot to mention is where I think that computation with quasi-stable transients, such as those that come from Gauss scaffolds, will be important when you have the time also part of the equation, where you need to remember something over a transient period of time, or you have to integrate over time, because this is these are the examples where you have to have this balance between robustness and plasticity. So we looked in the literature to see how or what is the description of about how neuronal networks encode or perceive time. And what we found in the references is that, of course, that this is very important for living systems, but what we found in the references is that how it is taught in neuroscience is that the timing is an intrinsic computation that emerges. So it's not a specific circuit in the brain, but rather it's an emergent property that, that comes from the inherent dynamics of the network. And how this is usually tested, very similar to some of the tasks that we have also seen yesterday. These are one of these very basic working memory tasks that they do in primates. So you give the monkeys a go cue, which you so the measure and the weight here, the first two signals. So the monkey has to remember this time interval. Then you wait for a delay period, and then you give them a go cue, and they should, for example, push a button exactly after the time interval that they have remembered. So we thought to train a minimal neural network, and in this case, we use gated recurrent units in order to be able to also, and we decided consciously for a very small network, and that is a 3-component network, because we wanted to know whether we can understand how the network encodes this information about the time interval. So you can train very efficiently this network to do the task. But what you can do, because this is now a very simple network, you can see how actually the dynamics of the network changes during the training. So you start from a single steady state, and during the different training epochs, what you see is that the system actually generates a bistable behavior. But at the end, when the training is finished, the system is poised close to the bifurcation. So this neuronal network, in order to be able to encode these complex tasks, it finds the solution that it's optimal to be close to a dynamical bifurcation. Yes?
So when you say training, you're changing the weight?
You're training, yes, the whole, yeah, we change, yeah. No, sorry. So it's a gated recurrent unit network. So each node is represented by 3 variables, which you can remap. It's a discrete system, but you can map it to a time continuous system. So basically, it's kind of activity of of each of the nodes. So then, because we are very close to a dynamic bifurcation, this is very similar to what we have seen before with the single cells. So we used exactly the same features what we have defined before. So this kinetic energy approximation in the network, where we see this cloud of slow points that the network has. And then in this cloud, you can also calculate the eigenvalues. This is the maximal eigenvalues. And we see that the system has only single fixed point, which we also saw from the organization of the network. And here we see this distribution of eigenvalues from negative to positive, which hints to the existence of that this cloud of slow points is actually a Gauss set. So when you look how the network works, actually, when we give the signals, so the trajectory continuously stays in this cloud of slow points. So when you give the signal, you move it away, it comes back. And actually, this time interval that the network has to encode and potentially reproduce, it corresponds to the distance that the trajectory travels in this cloud of slow points. So what the network does, or at least how we understand it for the moment, what it does during training is shapes this cloud of slow points. And then for different intervals that you need to remember, or different t delays, you just end up on different positions of this cloud of slow points. And this determines, or this is the way in which the network remembers this time interval. Again, the very, I think, interesting thing for us is that all these computations is done away from the fixed point. Just to remind you, the fixed point is in the upper right corner, and even after the network makes the prediction, it takes time for the system to reach the steady state. So it does not use at all this steady state for the computation. So we would like to think that both in biochemical networks, but in neuronal networks, especially for tasks that are related to time, that maybe the dominant mode of computation is these computations with the transients, which is important because trajectories do not settle on any attractor, which means that the system remains flexible, and they also evolve non-asymptotically because your landscape continuously changes under the influence of slow sets. So if the system stays on this set of slow points, it basically gives the system more time that it can use to integrate the information. So in this way, the system can remain both flexible, but it will be also robust, which is what one needs to be able to operate in an environment which is continuously changing. So of course, now that we know the principle, you can easily find a minimal biochemical network that can actually do the same task. So we think despite an analogy between how biochemical networks compute and how neuronal network compute, there might be a very generic principles that would allow to study also this more complex task on a level, or design experiments to study them on a level of single cells that we are actually currently starting to do. So just to wrap up, with what we did with these very simple experiments by challenging cells with dynamic signals in microfluidic devices, what we have seen is that they're not simple input-output machines, but rather they actively process and integrate the signals in the environment. We found, especially for the receptor networks, that it seems that it's a typical solution for many of these receptor networks, that they're organized at criticality. And this is important because it allows them to balance between robustness and plasticity in their dynamics, but by that also in the responses to the environment. And it seems also intuitive that if your environment is metastable, then better your intrinsic dynamics also operate in a metastable regime, because this is the way how you can respond to the environment. And one way how one can do this is by having these co-sets or more complex scaffolds, which seems are present in biochemical networks in single cells, but also in these models of neuronal networks that we have looked in a very simple examples. So this gives us some hope that we can actually extend the framework of learning also on the level of cells and be able now to ask questions how cells predict, anticipate, or how they time their responses. One example, as I showed you, this timing task, one can easily imagine such a task on a level of biochemical network in single cells. We also work on a more generic or global theoretical framework that will enabled to explain biological computations across the scales of organization based on computing with these ghost networks, but also to construct artificial neural networks or that have these ghost networks as the main computational capabilities, because we think you can have more minimal or small networks with small number of components that will be able to do perform the same task as many other RNNs. And with that, I would like to thank an amazing group of people with whom I have been fortunate to work with. The first work on the characterization of EGFR network and organization at criticality was the work of a PhD student, Angel. Abhishek and Achilles did the work on cellular navigation in changing environments. Then Achilles, Daniel, and Gayatri did the formal theoretical work on the ghost attracting set. The single cell learning is the work of Robert, and the RNNs is the work of Gayatri. Would like to thank an amazing group of collaborators with whom we have been also fortunate to work with throughout the years, our funding, and you for your attention.
I was curious, given that it has been shown that neural networks can be universal function approximator, do you think at some point we can also show that these ghost networks also can be a universal
yes,
function and then expect it to be cheaper kind of computation?
I hope. I think this is one of a line of research that we would like to follow up on. I think we still kind of struggle how to, I mean, we know how to construct like more simple ghost networks, but not in a more generic sense. So once we are there, then yes, that would be a next point to show.
Very nice. Thanks for the very nice talk. So a question about the task that you show that you have like the 3 nodes network, and then you have this delay. So just to make sure I understand correctly, so you have like this delay, and this delay varies. You can have different delays, and then you train a bunch of different delays, and just by doing this training, the network position.
Yes.
And this subcritical, like just close to the bifurcation.
Yeah.
And then does it generalize the different delays if you ask for a different delay?
So to some extent, it does. So you train it, let's say, on an interval between 30 and 180. So if you go out of this interval, it's better in the prediction the closer to the interval, and then you go further, the error is larger. So it does, but you can actually, it would work for any t and any t delay if you only change the amplitude of your go queue. So because this maps the system, you have during training, it sets this cloud of slow points. So what this amplitude of the go queue does is positions you in a different area. So basically you can do it for any t and any t delay without training. And yeah, and if you map it out, you get some kind of like an S-shaped function. So for any t or t delay, you have like 3 different possibilities how you can reach it.
Thank you very much. I'm just interested about, can you estimate this memory range of the cells?
So for these cells, so from theory, the closer you are to the bifurcation, the memory goes to infinity. And actually in those cells, because we have, we can measure the concentration of the receptor, which is how close you are to the bifurcation, we actually see that. The closer the system is to the bifurcation, the memory is longer. For this receptor, on average, it's 40 to 50 minutes. In some cells, it was even 150 minutes. For different receptors, you get different memory length. And we think that this is probably evolutionary, depending on the signals in the environment that the system has seen. but this is a speculation. So the memory length is different for different of these receptors.
Thank you very much.
So the length of the memory, at least in the model, so is dependent on the closeness to criticality. So, but there is probably a kind of optimal distance from the criticality depending on what the